Hi, my name is Melissa Hernandez, and today I will be presenting water jet, a simple method for classical conditioning in fish. Native Nile tilapia were used in this experiment. They are usually found in tropical and subtropical ranges such as Africa, the Middle East, and the Nile. However, they reside in freshwater habitats. For the most part, they have been introduced into the United States to be hybridized and introgressed in aquacultures. They obtain these fish from a hatchery and furthermore were juvenile when utilized in the experiment. The Nile tilapia was chosen since they are social and territorial. Furthermore, they have a high capacity for learning and memory. Now, some important definitions I need you to understand before I relay the rest of the experiment would be these key words. Classical conditioning is defined by Ivan Pavlov to occur in situations when a certain behavior, which is naturally expressed in the presence of a biologically relevant stimulus, is also able to be expressed in the presence of a previously neutral stimulus after being specific conditioning or training, which is labeled as a part of a learning process done in the subconscious. A conditioned stimulus is a stimulus that predicts the occurrence of another stimulus, which is always initially considered neutral, that can be detected by the organism, but does not initially induce a reliable behavioral response. An unconditioned stimulus is the predicted stimulus that can reliably induce a measurable response in the first presentation to the organism. An unconditioned response is a response that is not learned, but rather an innate or reflexive response to the unconditioned stimulus. Conditioned response is where the response is learned and is when there are repeated presentations of the conditioned stimulus followed by the unconditioned stimulus, where the conditioned stimulus begins to produce a conditioned response. Previous research shows how classical conditioning has been used to study animal cognition, especially in fish such as zebrafish, St. Peter's fish, and so on, where they have been found to associate stimuli as well as be able to express memory responses. However, in these kinds of research experiments, they utilized light or sound as a way of classical conditioning in order to seek the management of large groups of fish. However, research was limited on individual responses that took place in aquariums or small environments. Therefore, considering such measures of proximity in enclosed environments such as aquariums, there is a need for controlled experiments with more restricted conditions, since many errors have been found within utilizing light or sound as a stimuli. Therefore, in this experiment carried out by Adriana Barreto and colleagues, they decided to use localized movement of water by way of a water jet as a simple stimulus that would not interfere with other aquariums nearby. Here, they evaluate whether or not such a stimulus can be classically conditioned in Nile tilapia. As mentioned before, they used juvenile native Nile tilapia, and for 30 days prior to the experiment, they were assimilated in 500-liter tanks in order to make them accustomed to their new environment. The tanks were continuously aerated, used biofilters, and utilized PVC pipes to mimic shelters and to keep the pH balance at an appropriate level, as well as fed daily commercial food for tropical fish. Overall, a brief water jet was applied and followed by a food pellet located in the same place in the tank in order to condition the fish that were present in the tank. This procedure was repeated 4 times every day at random times throughout the day for 20 consecutive days. The conditioned stimulus, as defined earlier in the experiment, will be the localized water movement caused by the jet application. The unconditioned stimulus will be the food pellets, since food is considered a resource that animals are primarily motivated to reach. With each procedure, a latency response was tracked in order to see the response of the animals to the water movement. After 20 days, probe trials were performed in order to test the memory retention of the fish. Specific procedures taken place in the experiment where 10 fish were randomly chosen to be transferred to the experimental aquariums that were located apart from the maintenance environment they were assimilated to. All experimental tanks, each 5 centimeters apart, contained similar conditions as the maintenance tanks, but were smaller and used styrofoam plates for visual and physical separation. They remained on a shelf partially covered with a tarpaulin in order to make sure no other experimental groups were being influenced by other groups or by the researchers themselves. Holes in the tarpaulin were used to insert individual hoses that would serve as ports for the water jet and pellet insertion. The location of the hoses were randomized in each experimental aquarium in order to prevent lateration influences of fish from biasing a particular side. In the conditioning procedures performed consecutively, a water jet was manually injected into each respective hose using a syringe. After 10 seconds, a food pellet was introduced in order to ensure the fish would be conditioned to the water movement of the jet and not the pellet. Fish were then considered conditioned if they responded to the water movement right away on every test on certain days until the end of the 20 testing days. After these conditioning days were over, probe trials were performed where the fish would be tested on their memory retention on days 2, 4, 8, 16, and 32. The memory retention tested was used to see whether or not the fish responding to the water jet by association to the food pellet was maintained over a period of time. For these trials, which were conducted once randomly on those days, fish were exposed to the conditioned stimulus, the water jet, without receiving the unconditioned stimulus, the food pellet. After, the researchers tracked the latency response of the fish to the water movement. All of the statistical data analysis was explained and presented by the paper. Only mean and standard deviations were used in order to portray the data. This data, tracked on latency responses of the fish during condition tests, which took place 4 times every day for 20 days, resulted in the following. It was found by the study that at the end of the 20-day experimental conditioning period, all the tested fish were conditioned by the water jet. Looking at the data table for experimental days versus percentage of conditioned fish, the first fish was conditioned on the 7th day, and by the 10th day, more than half were conditioned. And on the 14th day, all of the fishes were conditioned. One can also notice on the learning curve to the right, constructed by the research paper, the error bars start to decrease as the days approach 220. Therefore, the fish had now been accustomed to the exposure of the controlled stimulus followed by the uncontrolled stimulus, meaning by the end of the trial period of all the fish were able to be conditioned by the water jet. As for the probe trials, fish responded positively since on each test probe day of 2, 4, 8, 16, and 32 days after the conditioning period, the response of the water movement caused by the water jet was always immediate for all tested individuals. Looking at the data and the results from the experiments, it is safe to conclude that classical conditioning was able to be performed on the Nile tilapia via localized movement as a conditioned stimulus and the food pellets as an unconditioned stimulus. This food pellet produces an unconditioned response of eating the food pellet, and after the classical conditioning, it helped produce a conditioned response of responding only to the water movement through repeated exposure to both factors. After all 20 days, all of the fish were able to respond to the stimuli as well as retain and use associative memory after the conditioning period, and up to 32 days, meaning the fish were able to retain the conditioned response to the water movement. This is an effective method for classical conditioning of fish, which can be used instead of light or sound stimuli. This method allows one to focus on a smaller collective sample of a certain species and control the conditions in a manner that is concise and controlled. This experiment helps prove memory retention in fish and is demonstrated in response to stimuli. The Nile tilapia are able to remember the localized movement of water from the jet and respond accordingly. The implications for this study show that simple water movement can be used as a method of classical conditioning. It is more controlled and safer than other practices. Thank you.